Hi there, I'm John, and I've taken on the rather foolhardy task of explaining to you about target screening technologies at Sigma in 5 minutes. I say that because I'm going to motor through these slides at a good rate of knots, but effectively when most people think about Sigma, they think about chemistry. These are going to be 4 reasons for you to think about as when it comes to target screening and target validation. So into the first one. If your target screening or target validation involves a cell line, we can basically make any cell line that you want from any background, with the exception, obviously, of human embryonic stem cells. We can do most things. So we produce custom cell engineering, I mean genetic engineer of that, for basic research, cell-based assays, target validation, drug discovery, and bioproduction. So, for instance, if you require a disease model with a patient-relevant knock-in or knock-out, we can make you an isogenic line. Basically, we can ship you the parental line and the line with the knockout. That will allow you to do a good, nice comparison. And we have various off-the-shelf models of this for colorectal, lung, and breast cancer. But also, if you're not interested in those conditions, if you're interested in another condition, we can make it as a custom for you. You may be interested in a fluorescent tagged receptor or biosensor. That is our EGFR biosensor cell line. That's available off the shelf. But if you want a different receptor tagged, we can do that. That's tagged under the endogenous promoter, so there are no toxic effects by overexpression. If you want a cell line derived from iPS, we can make you the knockout, and then you can differentiate that cell line down the particular pathway that you're interested in. You've then got a pool of cells you can work with. Or maybe you're into DMPK, and we have a new range of cell lines coming out for liver, kidney, and intestinal transport and metabolism knockout. We have the intestinal ones available now, but the liver ones will be released in the next month. So basically, we can do pretty much what you want, and we have customs and off the shelf, and we don't turn down many projects. So come and talk to us. We're at booth A3. The second reason. Knockout or knockdown. There are 2 papers there, and both papers are quite similar. They both describe screens for targets involved in tumour resistance in chemotherapy. The paper on the left is a shRNA screen. The paper on the right is a CRISPR-Cas9 gene knockout screen. They both identified very similar targets, but some unique targets were identified by the paper on the right in terms of knockout instead of knockdown. So some unique targets were identified by using knockout over knockdown. So we would say absolute knockouts can identify targets that may be missed by knockout screens and vice versa. And we're technology agnostic at Sigma. We have all these technologies for you to play with if you want to. So we have pre-designs for all human and mouse genes for both shRNA, for CRISPR-Cas9, and for zinc finger nucleases. So again, the costs are pretty similar. If you're interested, come and talk to us at booth A3. We also have a nice high-throughput screen for protein-protein interaction detection. This is Duolink. This is a technology we licensed from a Swedish company called Olink. We can't claim credit for it. But effectively, it's an antibody-based kit to localize and quantify protein interactions directly in the cell. It's a fast and simple protocol. You can use it in fixed cells and tissue samples. And it's suitable for high-throughput screening. We have a webinar recorded on our website where one of our customers at the European Screening Portal describes how he uses this technology in 384 well plates. That's a nice example of what you can achieve. I don't know if you can see, but this shows the demonstration of phosphorylation of the PDGF-beta receptor in response to PDGF stimulation. And if you can see the diagram on the right, each of those individual red dots is a phosphorylated receptor molecule that we've tagged with this kit. It's a very powerful technique. We like it a lot. So again, come and talk to us if you're interested. The final slide, another reason for you to come and talk to us is we can give you a whole genome lentiviral pooled shRNA library for under £10,000. You might think, what use is a pooled shRNA library to me? How do I deconvolute that? Well, basically, we'll ship you a pool of lentiviral shRNA clones in tubes. You can apply them to your cells, you can culture your cells, and apply them to apply a selective pressure. So you might employ a chemotherapeutic agent, you might sort them by FACS. At that point, you can then ship the pool to us. We will do the genomic preparation for you. We will do the next generation sequencing, and we will provide you data as to which clones in the pool have been enriched or depleted. And therefore, the targets that those clones were designed against can be implicated in the disease or biological process that you're interested in investigating. So, a lot of powerful techniques. Don't think of Sigma for chemistry. Come and see us at the booth. Thank you very much.